In this final hatchery module, we shall learn about the last steps for rearing and releasing Nephrops juveniles. At day 15, the majority of the larvae will have proceeded to molt into stage 3. At this point, larvae should be removed from the rearing tank and transferred to the post-larval rearing tanks. There are a number of options in configuration and design. Stage 3 larvae swim less and are negatively phototaxic. They are now increasingly cannibalistic, and therefore it's beneficial to separate individuals. After 21 to 24 days, the larvae will metamorphose to the first post-larval stage. The appearance is similar to that of adult Nephrops, except that they are around 15 mm in total length. Some larvae will not survive the molting process to post-larval stages and become entrapped in their old carapace. The post-larvae are now semi-benthic, meaning that they remain mostly attached to the benthos. They may have additional exploration time during the first 2 stages to find a suitable habitat, where the sediment is the right texture in order to construct a burrow or enter an already formed adult burrow. It takes 16 to 20 days for a post-larval stage 1 to molt into a post-larvae stage 2. The intermolt period increases in duration as the individual gets older. The Aqua Hive system was developed for Homarus gammarus for the development of larvae from stage 3 to post-larvae stages 1 to 2. The Aqua Hive plates, as seen here, allow individual larvae to be separated and raised in one plate. It holds around 350 larvae and has small holes on the bottom. This allows feed particles through the individual compartments in an upwelling tank. For Nephrops, it's best practice to place the plates within the rearing vessel and attach a second Aqua Hive plate on top with several elastic bands. The plate is submerged below the surface at around 10 centimeters. Animals are fed as normal with artemia and the plate removed and the filter taken off to allow uneaten food to flow out. Periodic monitoring of the development will be needed once a week, and removal of dead larvae and larvae metamorphosed to post-larval stage 1. Survival using this technique is around 40 to 50%. Alternatively, the stage 3 larvae can be raised in beakers or Orkney cells. The beakers shown here are set in a wet tray that allows temperature-controlled water to flow around the bottom of the beakers, thus controlling the temperature within the beaker. The beakers in the picture are 60 mm wide and 110 mm in height, and 1 stage 3 larvae should be placed in each. Again, the larvae are fed artemia at around 3 per milliliter daily. The beakers have no additional water flow or aeration and therefore need daily water changes. This is done by carefully draining the majority of the water out and then transferring the larvae into a clean beaker. The old beaker is then washed and dried, ready for another larvae. Survival for this technique is around 20 to 30% and requires more effort in terms of time, resources and space when compared to the Aqua Hive plates. Once the larvae have metamorphosed to post-larval stage 1 or 2, they can be transferred with the water-to-water method and placed into tanks with suitable substrate. The substrate has been shown to improve survival for on-growing when compared to the beakers or Aqua Hive plates. The post-larvae can construct burrows and feed on the substrate. The substrate should be collected from local Nephrops grounds to be sure that the substrate is the right consistency for burrow construction. The substrate can be collected with a grab. The mud is sieved through a coarse mesh of around 1 mm and then passed through a finer mesh of 0.5 mm. The mud should not be allowed to dry. The post-larvae are kept within beakers that have mesh exteriors to allow water exchange but keep the larvae separate. The sieved mud is placed in the bottom of the beaker and left for several days to stabilize. Several beakers are placed in a tray tank that has several water inflows at one side and outflows at the other. The post-larvae can be fed a range of food items, including pelleted feeds, artemia, and fresh or frozen seafood such as prawns. Larvae can be released at stage 3 or at the post-larval stage 2 onwards. In either case, the larvae should be released over current Nephrops grounds or where there is suitable substrate for burrow construction. Before the release, the Nephrops should be acclimatized to the ambient seawater temperature of the destined release site. Several release techniques are shown here that have been used for both Homarus gammarus and Nephrops. Diver-assisted releases are the most common and preferable, but this may not always be possible due to the increased costs and the limited depths that divers can reach for Nephrops grounds. The divers simply take several containers, which could be the Aqua Hive trays, and release the larvae directly onto the substrate. The Nephrops should be released at a density of around no more than 3 per square metre of suitable habitat. If the density is too high, there will be higher casualties due to increased encounters between the individuals and delays in finding suitable substrate and shelter. Surface release techniques are simply emptying the post-larvae out onto the surface and allowing them to drift down to the benthos. There may be increased predation during this method, but it's the simplest, cheapest and quickest method. Using release cassettes involves storing the post larvae in a device which could be, for example, the Aqua Hive trays or, as seen here, the Nephrops tubes. Paper that disintegrates slowly is placed over the top to stop the larvae from leaving. The cassette is lowered from the surface to the release site and the paper slowly degrades to allow the post larvae out. This method doesn't involve divers and can be used in conjunction with the local fishermen to restock fish areas. The release cassettes could even be placed within the creel itself.